All right, recording. All right, so our talk today is titled "Improving Tomato Flavor Using Biochemistry, Genomics, and Breeding" from Denise Tieman, who's at the University of Florida. She did her Ph.D. in horticulture at Purdue University, working on tomato also, and has been working on this tomato flavor in collaboration with Harry Klee for the last 15 years. So it should be really interesting to hear what they've been doing. And with that, I will turn it over to you, Denise, and let you get started.
Okay, great, thank you. Get this all set up here. Well, thank you very much for inviting me. I'll tell you a little bit about what we've been doing for the last 15 years on trying to understand tomato flavor. Well, the problem is that nobody likes the taste of tomatoes. If you look at this, this is just a little chart that tells you how big of a crop that tomato is. So 100 and so many tons here, a lot of tons of tomatoes. And the biggest producer is China, with the US being third. So as far as the United States is concerned, Florida is the biggest producer of tomatoes, and that's why it's so important that we do tomato research in Florida. California also produces a lot of fresh market tomatoes. So what is wrong with tomatoes? I think everybody knows what the problem is. Mostly is that people just don't like the flavor of modern tomatoes. They've all had a tomato from a farmer's market or grown it in their own garden, and they find that that tastes so much better than what's in the grocery store. So if you look at this, this is just how dissatisfied people are for different fruits. And And if you look at the red bar here, you can see this is tomatoes. And of all the fruits that are available, tomatoes have by far the most dissatisfaction from consumers. But you can see this is a problem with other fruits as well, where people just don't like the flavor of the grocery store tomatoes and would prefer tomatoes from a farmer's market or from their own garden if they have the chance to have a garden. So what can we do to fix this problem? Sorry, my mouse is a little crazy right now, sorry. Well, first of all, why are tomatoes dissatisfied? Why are consumers dissatisfied with tomatoes? And if you look, the top reason is that there's too little flavor, but there are other problems as well. Sometimes they're too soft or too expensive, which we can all relate to. Maybe they're too grainy or they're overripe. There's a lot of problems with the tomatoes, but by far the biggest reason that people don't like tomatoes from the store is that they have too little flavor. So how does this happen? Again, a tomato from a farmer's market or from your own garden often tastes much better, especially heirloom varieties are known for their good flavor. So why don't we see those in the grocery store anymore? Well, maybe you see them, but they cost $5 a pound or something, and that's not very practical for most people. So what has happened is the breeders have been breeding for the producer, for the grower of the tomatoes. And what the grower wants is disease resistance. Obviously, you want to get a good crop. You don't want them all to be lost to disease, shelf life, so that we can have tomatoes year-round in the grocery store. They need to be shipped long distances from Florida or from Mexico to get up north in the winter. And so you need a long shelf life. Yield is obviously important. What they look like, most people like a round red tomato. And uniformity, if they're all the same size, they fit in the crates much better, and for the shipping and everything. And so people like them to be a uniform size. So these are the producer priorities. And so what the breeders done is they looked at all these traits. They say, yes, we need these things. They develop molecular markers for instance, disease resistance, shelf life, and so on. And that's what they bred for. But what does a consumer want? If you look on the right-hand panel here, they want flavor most of all. Aroma, novelty. Sometimes these little packs of tomatoes with the colors, those are very popular. Something a little different. Nutrition, high lycopene, and sustainability. So how do we breed for the consumer? First of all, we have to have sensory analysis. We need to know what people actually like in a good tasting tomato. That takes a lot of time. You have to have a lot of people taste the tomatoes, chemical analysis to understand what makes the best tasting tomato, and then molecular genetics to develop markers and then to breed for flavor. So everything on this side, what the consumer wants, is much more difficult to breed for and to quantify than what the producer wants, especially the sensory analysis and the chemical analysis. It takes a lot of money and a lot of skill to be able to do this. They're not easy assays. It's very complicated. So these things have been left by the wayside as people have been breeding for new modern tomato varieties. So why is breeding for flavor so difficult? It's a highly complex multigenic trait. So there's many compounds that are involved, a lot of genes. We've only found a small portion of all those genes that are important for tomato flavor. It's hugely influenced by the environment. We have 2 seasons in Florida, a spring and a fall season. We find the flavor is very different during those 2 seasons. In this summer, when it's hot, when the tomatoes develop this flavor, and the fall, when it's cool, when it's developing this flavor, we see very different flavor traits. So that's another problem. So it's hard to quantify because it's so different depending on where you grow it. It's highly subjective. What I think would be a good tasting tomato may not be what you think is a good tasting tomato. Some people like them sweet. Some people like a more complex flavor. And again, as I said before, all these traits are very expensive to quantify. You need specialized equipment, trained personnel to look at these things, and it just gets really expensive. So it's not practical for a tomato breeder working in the field when he has to look at disease resistance and shelf life and so on. All these things are really important traits, but then you have to look at flavor, and oftentimes what the breeder would do is taste it and say, this tastes pretty good to me. And that's how the tomato has lost its flavor. So how can we fix this problem? This is where we started about 15 years ago. So what is tomato flavor? Most sugars and acids for sure. You want the sweet and the tart together. But what makes a tomato are more than 400 volatile aroma compounds. And these compounds come from many different pathways. And it's a particular problem for tomato because there are so many compounds. For some fruits, there's 1 or 2 compounds that really give you what's pretty close to its characteristic flavor. But for tomato, you need this whole mix of compounds, all in the right ratios to one another to get good flavor. So you're sensing the sugars and acids on your tongue naturally with your taste buds. But for the aroma compounds, what happens when you chew it in your mouth, it goes up through the back of your throat and up to your olfactory system in your brain, as compared to when you sniff the tomato, it goes through your nose to this same olfactory system. So here you're getting the aroma compounds, they're heated up, they're released, and then they go up through the back of your throat to your brain, and that signals this tomato tastes good or this tomato tastes bad. So how are we going to make it so it's easy to breed for flavor? The tools that we're trying to develop, first of all, we had to determine naturally which are the important volatiles that drive consumer preferences. You can't practically quantify 400 compounds and breed for all those 400 compounds to have them in the right amounts in the tomato. So we need to figure out what's the most important volatiles first of all, and we'll start focusing on those first. Then we wanted to define the genes controlling their synthesis, and this is the biochemical aspect of the whole project where we actually are defining a lot of these biochemical pathways. Sorry, what happened here? Oops, no, sorry, I messed up here. Let's see my whole talk here. Let me just exit. Sorry about that. Yeah, so the biochemical aspect of this is that we don't know all the pathways to these compounds. As a matter of fact, when we started, there were very few of these biochemical pathways that had been defined. So we're working on that from a basic science point of view. And then what we wanted to do was to identify the best alleles of these genes. And then if we can reduce these traits to molecular markers, then we can breed for better tasting tomatoes. So what we've done is to start this project, we needed to look at a lot of different tomato varieties and look at the biochemical attributes of these. So we quantified sugars, acids, and the aroma compounds from many modern varieties, heirloom varieties that presumably had good taste, and also some of the early domesticated tomatoes, which we did in collaboration with Esther Vandernap's lab at the University of Georgia. So, well, this is just a little bit to tell you exactly what we did. We quantified about 60 or more flavor chemicals in about 700 now varieties we've done. And we measured consumer preferences on a fraction of those. With these, we did consumer panels where we asked about 70 to 80 people to taste each tomato variety and rate them on how much they liked them, sweetness, sourness, and so on. And then we developed statistical models to explain which of these compounds were most important for flavor. Do we need to increase it or decrease it in a tomato to make a better tasting tomato? And then we also identified those targets for metabolic improvement. So why volatiles? Obviously we can increase the sugar, make the tomato sweeter, but that also gives us a problem with yield, because usually when the plant puts more sugar into the tomato, the yield is usually decreased. So we decided to focus on the volatile aroma compounds because they're active at very small levels. So to increase them does not really give you a metabolic hit where you decrease the fruit weight. They're essential to good flavor. So a tomato with reduced volatiles and still has the sugars and acid, it tastes very bland. So if you put a tomato in your refrigerator, you start to lose the aroma compounds, and that tomato doesn't taste as good because you just have the sugars and acids. They're still there as a constant, but you've lost the aroma compounds. And again, also, we don't want to decrease yield because that's what the growers need. So this is a lot of biochemistry here, but these are just some of the compounds that are important for tomato flavor. If you look here, these actually come from many different biochemical pathways. But if you look at these two columns right here and this column here, it shows you the huge variation that you see in these compounds in different tomato varieties. So we've grown, like I said, over 700 of these. And you look at this, you can have up to a 3,000-fold difference from one variety to another in the amounts of these compounds, which is pretty amazing. If you look, these are the flavor descriptors of these. So for instance, methyl salicylate smells like wintergreen, and 2-isobutadiazole here smells like the tomato vine. And but if you look at this, none of these says tomato, just tomato, you know. So tomato flavor is really this very complex combination of all these different flavor characteristics. This is some work we did in collaboration with Marcio Resende's lab at the University of Florida. And if you look at this, they developed statistical models to tell us how much each of these compounds contributes to overall consumer liking. So this is with the taste panel data that we have, where we had so many people taste the tomatoes. And if you look at this first here, this is overall liking. And you can see about 43% of the contribution to the overall liking is from sugars, 9% from acids. But if you look at the aroma compounds, about 42% of the total overall liking is from the volatile compounds. So these are obviously very important. You need the sugars and acids, but you also need the volatile compounds. This is actually, sorry about that. This is actually Sweetness, this is the contribution to the sweet. So people were asked, how sweet is this tomato? And this is the data from that. And you can see to sweetness, the volatile compounds actually contribute 62% as compared to the sugars and acids. So we also know now, we've done a little work with this, and we show that if you have higher amounts of some compounds in a tomato, it actually makes that tomato taste sweeter without increasing the sugar amounts. And the last one is overall flavor intensity. So not necessarily how much you like it, but the tomato flavor intensity. And you can see the volatile compounds are truly important for tomato flavor, where 68% of the flavor intensity is coming from the aroma compounds and only 31% from the sugars and acids. So why are modern tomatoes, don't they taste good? Well, this graph really shows you what has happened over time as they bred modern tomatoes. If you look at this, what it's showing is the decrease in the amount of this compound in an average of all the heirloom tomatoes as compared to the modern tomatoes. So in modern tomatoes in general, the levels of methionine are about 60 to 70% less than they are in overall heirloom varieties. So if you look at this, almost all the compounds on this list are actually decreased in modern tomato varieties, and with just a couple down here that are actually increased. So we've lost flavor as we bred modern tomatoes for these other traits. So this just shows it from a genetic standpoint. This is a tree of over 800 tomato varieties now, including some wild species. But if you look at this, the fresh market tomatoes are this very narrow part of the tree. So our germplasm is very narrow. If you look at the heirloom tomatoes here and then the early tomatoes from South America, you can see there's so much more diversity there than there are in the modern ones. So we've lost all this diversity over time as we've bred for other traits. So we've done a lot with the biochemistry as well. And early on, we used a lot of introgression populations to identify the biochemical pathways to some of these compounds. And this just shows you a few of the different things that we've used. Pimpinellifolium, obviously, Cheesemanii, Haberchides, and Pinellii were some of the introgression line populations we used to identify genes in these biochemical pathways. And this is where we got, so this is what we know so far for the biochemical pathways to a lot of compounds. It's a lot of biochemistry. But if you look at this, everything in red is an aroma compound, a volatile compound from tomatoes. And the black arrows, the solid black arrows are where we know something about the biochemical pathways. Sorry about that. We identified the genes there and showed biochemically that these are the enzymes that catalyze these different reactions. If it's a dashed line, that means that we don't know the enzyme that actually catalyzes that reaction in tomato. So the orange boxes are the names of some of the genes that we have identified. And you can see that we have different pathways here. So these compounds here come from phenylalanine, and these have very floral scents. 2-phenylethanol smells like roses. Phenylacetaldehyde smells like hyacinths. If you look here, these two compounds are actually considered negative for tomato flavor, methyl salicylate and glycol. Methyl salicylate smells like wintergreen. This is a section of the pathways that we don't know a lot about. We haven't identified a lot of the enzymes, but we have some possible ideas of what the enzymes would be, but we have not identified in tomato. These particular compounds here have kind of a musty smell, and they're very common in cheeses and beers and things that have a lot of fermentation going on. This part of the pathway comes from lipids. So these all come from lipids, and these give you the green grassy notes in tomato. And we know quite a bit about these pathways. These here are the carotenoid derived compounds. So 6-methyl 5-heptene 2-O is derived from lycopene. the red color in tomatoes. And these have very fruity floral scents, and in general, they're considered very important for tomato flavor. Beta-inon is an interesting compound because a lot of people can't smell it. It's supposed to have a very fruity floral scent, but people like me can't smell it at all, so it's not adding to the tomato flavor. And this just shows you how different people sense tomato flavor in different ways. So like I said, we've identified some of these biochemical pathways, but now we can use genomics tools to continue to identify more of the pathways and to better understand tomato flavor. So at the point when we had a lot of biochemical data, genome sequencing became affordable, and we went ahead and we sequenced about 600 tomato varieties with in collaboration with Esther Vandernaps and San-Wen Huang's labs. And then we also looked at all the flavor chemicals in those same varieties. And then we used that to identify genes and also to make markers for breeding better tasting tomatoes. So this is just a little bit of GWAS data, and it shows some of the loci that we have identified. On the right, we have some sugars loci, and you can see for glucose, fructose, and soluble solids, we actually seem to identify those same loci on the bottom of chromosome 11. So along the bottom is the chromosome number, and anything above the red line is considered significant. So on the bottom of chromosome 6, there's a locus that seems to affect citric and malic acid. And if you look at this, the panel on the right, the volatiles, here you can see on the bottom of chromosome 4, there's a locus that affects multiple phenylalanine-derived volatiles. So I can use these GWAS information to try and identify new genes in these biochemical pathways and also regulatory genes of some of these pathways. So this just again explains some of the tools that we're using now to find causative genes. So we have the reference genomes. We have multiple high quality reference genomes within just the last few years, especially from Zach Lipman's lab at Cold Spring Harbor. He's sequenced several varieties with really high quality reference genomes. We have the Illumina sequences of over 600-700 varieties now. We have RNA-seq data from some of these, and we also use gene annotation to try and understand which genes could be possibly catalyzing what reactions. So I'm just going to go through a couple of these and how we've gone through to identify some of these genes and enzymes that are important in these pathways. This is a locus on chromosome 9 at the bottom of chromosome 9 that affects methyl salicylate and guaiacol, which are actually both negative for flavor. In general, people like the varieties with less of these compounds. This is some work from Zach Lipman's lab as well. When he got these high quality reference genomes, he could start to look for structural variations. So another group has identified this gene. They call it non-spokey glucosyl transferase. If you have the wild type version of that gene, it actually adds a third sugar onto a 2-sugar conjugated guaiacol molecule. And this cannot be converted back. Sorry. This can't be converted back to guaiacol. But if that gene is knocked out, you have a disaccharide attached to the guaiacol molecule. This can be converted back to guaiacol, the compound that you can smell. So if you look here, there's a lot of structural variation going on here. In the original Heinz reference genome, there was actually a stretch of ends here originally. So these high quality genomes have helped a lot. You can see the first haplotype has 2 copies of this gene that are active. The second haplotype has this first gene knocked out, and 1 active copy of the gene. This one, haplotype 3, has knocked out this gene but added a new copy. So it has 2 functional genes. Haplotype 4 has no functional genes, as well as haplotype 5, which has a huge deletion that deletes the whole locus. So if you look at the levels of guaiacol in the different varieties, so this is just an average of all the varieties that we had. And we looked at the different haplotypes. So the ones that have the functional versions have very low levels of guaiacol, while the 2 that have no copies of the gene that are active have very high levels of guaiacol. So this is a new thing that we can use now as the structural variation to understand why we're getting all these different levels and such huge variation in some of these compounds. So let's go on to another one. This is one that we have, that was done in our lab. This is a methylesterase on chromosome 2. So there was a GWAS locus here, kind of in the middle of chromosome 2. And we started to look what was around there. And we found 4 copies of a methylesterase on chromosome 2 in this region. And we start to look at this and we again see a huge amount of structural variation. And we have 8 different haplotypes that we've seen in all the varieties that we have looked at. And what you can see here on this panel down below is that haplotype 2 has much higher levels of methyl salicylate, and also haplotype 4. If you look at these both have this insertion before in the promoter of the first gene there. The ones that are in light blue probably do not have activity. The ones in pink also do not have activity. So you can see that we're again seeing huge amounts of structural variation and differences in activity. And so we see different amounts of the volatile. So Libby Frick in our lab, she actually generated CRISPR knockout lines of this. And you can see this is the wild type, and the knockouts have much higher levels of methyl salicylate. And this is what the enzyme's doing. It's taken methyl salicylate and converting it to salicylic acid and methanol. So methyl salicylate is the volatile compound. Salicylic acid is not volatile, so it wouldn't affect flavor to the extent that methyl salicylate would. So you knock out this gene, you get higher levels of methyl salicylate. So we've learned a lot about the biochemistry, but we want to use this for a practical purpose. So now we would like to begin to be able to make it easy to breed for tomato flavor. So we've identified quite a few GWAS loci. If you look at the red dots on this slide, you can see where we have different GWAS loci that usually affect multiple compounds and in the same pathway, and from many different pathways. And we do want to take care that we don't use loci that are really close to some kind of agronomic trait, for instance, fruit weight or disease resistance. So we've chosen some of these that are not too close. Something like this we wouldn't want to use just because it's so close to some other important traits. So we chose some of these loci and we're starting to breed them back into a modern tomato variety. So for instance, this is an allele, GWAS loci on chromosome 9. Sorry about this, I'm having an awful time. But you can see this is just the average of all the varieties that we tested that have the reference allele that are heterozygous or the alternate allele. You can see anything, the average of the alternate allele is much less. If we look at the SNP at this locus, the alternate allele has much less methyl salicylate. So this, since methyl salicylate is negative for flavor, we would very much like to get the alternate allele into a modern variety.
Sí, o sea, yo podía ir a activa sin ningún problema.
Claro.
Somebody's got their microphone on. If you could silence that, please.
So, as I said, tomato flavor is complex, so we can't just add in one locus that's going to be important for one aroma compound, because that's not going to do much for flavor. We need to add in a lot of different compounds. So we chose some of the loci that had the biggest effect on different compounds. If you see the compound in a red box here, this is something we want to reduce the amounts of. So these two we want to reduce the amounts. The other compounds we want to increase the amounts of. And we identified GWAS loci that had a big effect in these particular compounds. And we what we wanted to do then is to put these into a modern tomato variety. So unfortunately, you're not going to find all of these in one heirloom variety. So we had to start with a whole bunch of different ones. So we started with several here: Maglia rosa, brandywine, red currant. And we took different loci from these parents, and then we started to combine all these into a modern commercial tomato. And for some of these loci, we don't actually know what the causative genes are, but they came up in the GWAS, so we're going to use them even if we don't know the causative gene, which is nice because it saves us some time as far as the breeding project is concerned. So we introduce a bunch of loci into these, and then we want to know, if, first of all, if people can taste the difference. So what we do is a triangle test where we give people three different samples, two of them with one genotype and one with another genotype, and we randomize these for different people. So if you see here, we just put them in different orders, and some people get two of one variety and some people get two of another variety. And then we ask them to identify the odd sample. So they'll tell us which one of these is different, basically. And these are the results of this, and this is a little complicated because the statistics are a little different. But if you get the correct responses are the black bars and the incorrect responses are the white bars. But if you're above this red line, it means that people statistically can tell the difference. And as you can see, with a bunch of different loci where we've added different aroma compound loci. So this one had geranyl acetone and 6-methyl-5-heptene, 2-O and higher in these, people could tell the difference. This one had 3 different compounds, and people again can tell the difference, and so on. So with the many different combinations of these, people could tell the difference. The one they did not tell the difference was this particular combination here, where it had higher 3-methyl-1-butanol and reduced glycol. So now we had just a few loci in those, so we start to stack these traits, and this is just our breeding scheme. It's been quite a few years since we started this, with a lot of back crosses to get everything. So we have these loci in mostly our commercial parent background, the Florida 8059, which is something from the UF breeding program that we started with. So we start to combine loci, and this is these particular lines had 4 or 5 loci combined. And if you look at them, this is our control, Florida 8059. And so far, we only have one variety that is significantly better. So people said this one tasted better than Florida 8059. So we still have some work to do. So this is 4 or 5 loci. And now we're to the point where we're going to combine even more loci. In the summer, we'll have some with 6 or 7 loci that we're going to test. And we really hope that we'll have more lines that have better flavor. And so we're very excited about that. But at least now we found one line with a specific combination of loci that actually has improved flavor. So the bad news is that most growers won't accept better flavor if it comes at the cost of yield. They're paid per pound, and that's what they need, is they need a huge yield to make a profit. So the good news is that we know how now to breed for better flavor, but it's going to take us some time. just because there's so many back crosses there, to keep all the agronomic traits and to introduce enough flavor loci to give us better flavor that people could actually detect. So while we're waiting on that, we've decided to do some stuff for the home garden market, because home gardeners don't care so much about size and yield and the ability to ship the tomatoes. So what can we do? We've made hybrids of a commercial variety with some really good tasting heirloom varieties. And these are the 3 varieties that we've released so far: Garden Gem, Garden Ruby, and Garden Treasure. And people really like this. We hear from a lot of our home gardeners, and they really enjoy the flavor of these tomatoes. This is just our Facebook page, but so far we've provided seeds of these particular varieties to about 15,000 different home gardeners in all 50 states and 40 countries. So this past year, we launched what we call our Citizen Science Initiative, in which we actually ask our home gardeners to collect data for us and to rate these tomatoes. So they're collecting data such as yield, fruit size, flavor, obviously, and how well the plants grew in their particular environment. And this has been hugely helpful for us, because we have people growing them all over the country. So these are just some of the places where people have grown these varieties for us. Florida obviously has a rather extreme climate, so growing them only there, we only get so much data of how they perform. So having people send us data back from all over the country has been hugely helpful for us. And I think we sent out about 700 packets or sets of seeds. And I think about 120 people sent us data back. So this is the data that they sent us. And if you look at this, they rated it for flavor, overall performance, yield, and appearance. We actually have one RC hybrid here, which scored really well for flavor and performance for all of these things. And Garden Gem also, and even the Brandywine. And we sent them Better Boy seeds as a control. And I was actually kind of shocked how poorly it did. You can see it was the lowest rated of any of the varieties. So this was really great data for us. We know our varieties taste good, and they actually did much better than the Better Boy, which is kind of a standard for home garden tomatoes. So all in all, what we've done is we've defined the chemistry of a good tasting tomato. I've identified loci that are important for good flavor, and we are close to significant flavor improvement of a modern commercial variety that hopefully we can make it easier for breeders to actually breed for tomato flavor. We have released our heirloom hybrids, which home gardeners love. And we just hope that everybody can have a better tasting tomato from the grocery store sometime soon. And I'd like to thank a lot of people. First of all, at UF, Harry Klee, obviously, Shang Li, who did a lot of the structural variant work, Libby Frick, who did the methyl salicylate locus, Modesta Bugo, who has done a lot of the breeding work, and Marcio Resende, especially, who's helped us with a lot of the bioinformatics, and also Patricio and Felipe were working with him. Andrew Hansen, who's helped us with a lot of the biochemistry, and Charlie Sims and Linda Bardashek, who helped us a lot with the taste panels. And from other places, especially Esther Vanderknapp at the University of Georgia and Anna Casedo at the University of Massachusetts, have been very instrumental in helping us with the bioinformatics and with a lot of other stuff that goes beyond the biochemistry that we do in our lab. And Laura and Manoj and Hamid, who are also in their labs. Alice DeFerny, who helps us with a lot of the biochemistry. Jim Giovanoni, who's done a lot of the genomic stuff, and also Zhangyan Fei, working with him. Sanwen Huang and Zach Lipman, who did a lot of the genome sequencing. Thank you. Questions?
Hey, thanks. That was a great talk.
Sorry about all the trouble with the slides flipping.
Oh, no worries. Yeah, it's always, Zoom's always an adventure.
Yeah, it sure is.
So everybody, if you have questions, you can either type question or Q in the chat, and then you just call on you and you can ask, or if you want to type the whole question in the chat, I can read it. So just either, whatever your preference is. It looks like Simon, you have a question?
Yeah, thanks a lot. Thanks, Denise. That was really, really exciting. So in one of your slides, you showed how you sort of avoided QTLs or loci close to agronomic traits, such as, I think, fruit size. Is it, I mean, I understand the motivation for doing this in your context, but is it known in general whether the good I mean, one could imagine there being good combinations at those regions where this linkage now potentially is disadvantageous. Do we know whether the good varieties are still around, or have we completely lost those, or did they never exist?
Oh, you mean combinations where they might have large fruit and another trait at the same place?
Yeah, exactly.
That's a good question. I don't know that we've actually looked at that necessarily. There are very large fruited heirloom varieties, and I've not looked to see, if a particular locus next to that has been, has the good locus or not. And that's not really something I've looked at, but it's an interesting question. Yeah.
Thanks a lot.
Mm-hmm.
Anybody else questions? I have a question. I was kind of wondering when you were showing all those pathways and all the different classes of molecules that are involved in the flavor profile. I was wondering, how much is there a potential for like more sort of higher ordered interactions between all the compounds in the profile? That like is the way that a person perceives one of those things dependent on the concentrations of all of the others? Does that make sense?
Yeah, I mean, it's a very hard thing to determine experimentally, but just from knowing the data, it's interesting because methyl salicylate's very much a negative for tomato flavor, but we actually find high levels of it in some heirloom varieties that have excellent flavor. So it seems, if you have other compounds, higher levels of some of these other compounds, it doesn't matter that you have something more of a bad compound just because the other compounds are making it taste good anyhow. So yeah, it's something, experimentally we've not looked at, but it definitely has been happening. And it's interesting, another negative for this flavor are these acetate esters. But again, we see that in a lot of these high levels of that in heirloom varieties, but people still like them. But again, the acetate esters are interesting because in a lot of different fruits like melons and things, they're very important for flavor. They're very positive for flavor, but for tomatoes, they're a negative. So it's interesting how, different fruits, maybe you have different expectations, but also, it's probably the combinations of the other compounds that are there that make you say, oh, this tastes good or this tastes bad.
Interesting.
Yeah.
Federico, you have a question?
Yes, a very nice talk.
Thank you.
I have a question which is likely obvious to a breeder, but so you have found a relatively small number of loci with very strong effects. Why did you choose an approach of doing traditional breeding, like a lot of back crosses, rather than transgenesis? Is the main reason that people don't like transgenics, or that biologically you
expect the reasons, but we can do CRISPR now here in the US, and if you do enough paperwork, they're not considered transgenic. But the thing is, with CRISPR, you can only knock out genes. So, for a lot of these, we want more gene activity rather than knocking them out. But yeah, I mean, we're trying to make it so people can breed for it. But would be nice to be able to make transgenics and actually show that you can make better flavor that way. Maybe it would convince people it's not a bad thing.
But is there maybe a biological reason why maybe by doing backcrossing you might expect less negative pleiotropical effects or something like that? Or is just maybe a marketing reason rather than...
Yeah, I mean, definitely we could get some problems with it. But again, I think we just this is our first try at breeding. So, you know, this is basically a test case. Can you breed for flavor? So this is our first pass, and we may have to modify things at some point in the future to, you know, get even better flavor. But as a first pass, at least we seem to be getting it so people can detect the differences. And we have one particular line that actually people rated as better. So I'm very hopeful that it is going to work. But I do think it'll be a long process to be able to breed for the perfect tomato flavor. Well, the other problem is my perfect tomato is not your perfect tomato. So.
Yeah, that's another problem. Okay, thank you very much. Very nice talk.
Hi, I have a question.
Oh, hey, Jake.
Hi, Denise.
Hi.
So I know that some of these volatiles come from the carotenoids that give the tomatoes their color.
Mm-hmm.
So can you say that a tomato with a very bright, vivid color will have better flavor, or is there not a direct connection there?
Well, yeah, it may have it'll have a different flavor for sure. Yeah, we know that 6-methyl-5-heptene 2-one is very important for flavor, and that comes from lycopene. And we do, in general, depending on the color of the tomato, we'll see differences in the levels of these carotenoid-derived volatiles. If you get these yellow, really pale yellow tomatoes, they won't have much of the carotenoid-derived volatiles, and people can definitely taste the difference and do not like them as well. So you get the orange ones where they have a lot of beta carotene, they'll have more beta iodine, and some people like that and some people don't. So you're switching the carotenoid derived volatiles that they'll have. But again, I said a lot of people can't smell beta iodine. So for me, I probably wouldn't be able to tell the difference, but other people would.
Very cool. Thank you.
You're welcome.
All right, any other questions? If not, I guess thank you very much, Denise. That was a really interesting talk, and we'll get it uploaded to YouTube pretty soon. The next seminar is going to be, not next week, but the week after, and it's a graduate student from the Federal University of Rio Grande do Sul in Brazil, Alexia. Let me pull up the schedule so I can tell you the title. It's going to be investigating characteristics of pollination attraction in natural hybrids between petunia species. It's Alexia Gonçalves Pereira, and so that'll be next time, and that'll be the last one for the semester. So anyway, thanks again, Denise. That was really nice, and thanks everybody for coming. All right. See you all next time.
Bye-bye. Thanks for having me.